Epigenetics refers to the heritable modifications in gene function or activity without changes in the DNA sequence. Epigenetic processes are genetic modifications that affect gene regulation by changing the DNA conformation. Epigenetic changes are found in many different cancer cell types. One such change is histone modification that may be caused by overexpression or aberrant recruitment of HDACs. Within the chromosome, DNA is packaged into chromatin. Chromatin consists of DNA, structural histone proteins, and non-histone proteins. Within chromatin, the repeating unit is the nucleosome. Nucleosomes are made up of 146 base pairs of 2 superhelical turns of DNA wrapped around a core of 8 histones. The histones are responsible for maintaining the chromatin's shape and structure. Epigenetic modifications, such as histone acetylation, occur at the amino terminal tails of the histones that protrude from the nucleosomes. Acetylation of histones is generally acknowledged to play a key role in the regulation of gene expression. Histone acetylation is controlled by the balance in the activity of 2 enzymes, histone acetyltransferase, or HAT, and histone deacetylase, or HDAC. For a gene to be transcribed, it must become physically accessible to transcriptional machinery. Acetylation of histones by HAT causes uncoiling of DNA and an open chromatin structure. This causes genes to become accessible to transcription factors, allowing gene expression to occur and proteins to be made. Conversely, deacetylation of histones by HDAC results in tight coiling of the DNA and a closed chromatin structure. In some cancer cells, there is an overexpression of HDACs, an aberrant recruitment of HDACs, or an underexpression of HATs, resulting in hypoacetylation of histones and therefore a condensed or closed chromatin structure.
There are 4 true stages of the cell cycle: G1, S, G2, and M. A 5th stage, G0, is where the cell remains indefinitely until it begins the cell cycle again. Some cells may never leave the G0 stage. Others that go through division rapidly, such as bone marrow cells, may skip the G0 stage entirely. Cells begin the cell cycle by entering interphase. In interphase, the cell prepares for division by duplicating DNA. Interphase includes the stages G1, S, and G2. In G1, the cell synthesizes DNA and protein and increases organelle and cytoskeletal elements. The chromatin consists of long, slender rods jumbled together in the nucleus. S is the synthesis stage. Nuclear DNA replicates along its centrioles, which are small structures made up mostly of microtubules that associate with the chromosome's DNA. The chromatins begin to coil so that they shorten and thicken. In G2, RNA and protein are synthesized. Proofreading also occurs. The DNA replicated in the S phase is checked for mistakes. If mistakes are found, the cell repairs them or receives a signal to self-destruct called apoptosis. The difference between G2 and G1 is that in G1, DNA and protein are made, while in G2, RNA and protein are made. In the M stage, the cell divides into 2 identical cells through divisions of the nucleus, mitosis, and cytoplasm, cytokinesis. Over 5 different stages, the centrioles pull the chromosomes apart and the cell divides in 2. First, in prophase, the duplicated chromosomes appear and then split into identical halves called chromatids, attached by spindles called centromeres. The nuclear membrane disappears and spindle fibers form in the cytoplasm, radiating from the 2 centrioles located at opposite poles of the cell. In metaphase, the spindle fibers pull the centromeres, aligning them in the middle of the spindle or the equatorial plate of the cell. The 2 chromatids are clearly visible in this stage. In anaphase, the centromeres split, pulling sister chromatids to opposite sides of the cell. By the end of anaphase, 46 chromosomes lie at each side of the cell. When chromosomes are separated, each sister chromatid is considered a new chromosome. In telophase, a nuclear membrane forms around a group of 46 chromosomes. The spindle fibers disappear and the chromosomes uncoil. Cytokinesis occurs and the cytoplasm divides into equal parts. Two identical daughter cells have been formed. Each new cell moves back to the G0 stage and is ready to begin the process again.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug-of-war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.